are specific to the particular pathogen. Now, it's also called therapies where you use antibodies is also called serum therapy, and it was first used as far back as the 1990s. It tends to be effective to have to be administered very early on in disease, but it can show a lot of variation. So serum therapy initially, before we knew how to produce antibodies, was really where you took blood and you isolated the antibody. So this was blood from somebody that was infected with the disease or had the disease. So you take blood and you isolate the antibodies from that donor. Those antibodies are purified, and then you can treat somebody else which gets the infection with those antibodies. However, I have kept going on and on about these B cells. So this does not give you immunity. And this does not give you immunity to subsequent infections, because antibody treatment obviously does not produce an immune response. So you do not get these memory B cells formed. So you are not immune from subsequent infections. However, it can be used to treat that particular infection. So it doesn't give you immunity, but it can treat disease. Now, there's a lot of variation, and the reason for that is obviously that you might have a different amount of antibodies produced in different individuals, etc., etc. Now, today, we still actually today do use this concept of passive immunity, where you take, I'm going to show an example of where it was used recently, where you take and isolate antibodies for somebody that's infected and then inject them into somebody that has got been infected with the disease. However, for many things today, we also produce monoclonal antibodies. Monoclonal antibodies obviously solves that some of that variation and availability that you might see if you're relying on humans. So just here is actually of the serum therapy. So it was actually, remember we talked about the Ebola outbreak recently in Africa in 2014. Well, in the Ebola outbreak, they actually did use serum therapy for some patients. So they took blood from an Ebola infected person or a survivor, and they injected that into a patient. Now, another place where you would often see this serum therapy used is as anti-venom. So in response to snake bites, somebody's been bitten by a snake, you might, or they have stored antibodies from somebody that has survived a snake bite, and you can then get those antibodies. Now, monoclonal antibodies, you might not know what that is, but that is shown in the slide here. So monoclonal antibodies is produced by injecting an antigen into mouse. The mouse then produces B cells. Now, these B cells is then fused with myeloma cells. So myeloma cells is the cancerous cell line. These myeloma cells will integrate or fuse with the B cells, and because it's a cancerous cell line, they proliferate very fast, and the cells produce antibodies. So you produce these hypodoma cells consisting of a fusion between myeloma cells and B cells, make monoclonal antibodies. And these monoclonal antibodies, which are made, are obviously depending on the antigen that you are injecting into the mouse. Now, one problem with monoclonal antibodies is that they can induce a response, an immune response, when they're injected into human. So it basically means that the antibodies are eradicated by the immune system. To avoid that, sometimes you do still use monoclonal antibodies for treatment of human diseases, which we'll talk about in a minute. But you can have, just depends on the antibody. You can have antibodies, so antibodies are developed which you can't use because they're just eradicated by the immune system. So you'll never get delivery of the antibody, which is high concentrations, which are high enough to be effective. Now, what you will see a lot now is that monoclonal antibody has been humanized, so the sequence of them have been slightly changed to look much more like human antibodies rather than monoclonal antibodies. Okay, so here is an example of when monoclonal antibodies are used. There is a lot of monoclonal antibodies which are now approved for treatment of a variety of different cancers. So when antibodies are used for cancers, they're called, or they are typically, what is called tumor-specific antibodies. So these are antibodies which are raised against antigens that are often overexpressed or only expressed. It's quite rare you see something which is only expressed on cancer cells and no other healthy cells. However, you often see overexpressions of many proteins on cancerous cells. Now you can then raise an antibody against those antigens which are heavily overexpressed on the cancer cells. And then you can treat the patient with these antibodies. Now these antibodies will obviously then bind to that antigen on the cancer cell. And remember how we talked about that the antibodies have got this common region. So they have got the variable region, which is the very specific region. So the variable region is the one which binds to the antigen. It recognizes very specific antigens. So it binds to the antigen. But we've also got the variable region, which is here. And that variable region, some of the, as we talked about yesterday, some of the immune cells have got receptors and can bind to that region and become activated. So in the example you see here is an actual killer cell, which is binding to that region. It could also be other cells, as you know, as we talked about yesterday. Now, it is actually often not only depending on the immune system, these treatments, because very often some of these antibodies are raised against receptors, for example, which are overexpressed in cancers, and they often play a very big role in the growth of the cells. So, for example, a good example is antibodies against the epidermal growth factor receptor. So the epidermal growth factor receptor is highly overexpressed in a lot of cancers, actually. Now, you can, there's antibodies against that receptor, and the epidermal growth factor receptor, when that one is activated, it leads to a signaling cascade which increases cell proliferation. And obviously, you know, cancer is excessive cell proliferation. So not only can the antibody activate the immune system to get rid of the cancerous cells, but actually the antibody can also block a signaling cascade which prevents the cell from proliferating. So it's kind of got a double effect, activating immune cells to get rid of the cell, but also blocking pathways which are really important for cell growth. So you can get a double effect with some of these antibodies. Okay, treatment with T cells. This is also something which is used to treat cancers. In this particular case, it is T cells which are isolated from the patient itself. T cells are then modified in a tissue culture dish, and then they're given back or injected back into the patient. It's called personalized medicine. We're going to look a little bit closer at it now. Now, before we look at it, where this treatment is very useful is in tumors where the immune system is not recognizing cancer cells. Okay. Okay, so this is what happens. It's also called CAR-T cell therapy. So it's chimeric antigen receptor therapy. Now, so what is happening here is that you take a blood sample from the patient, so here, you isolate the T cells. In a tissue culture dish, you then genetically modify these T cells to express two things. One, the outer region of the T cells, so it's a receptor, should have said that first. So basically, you transfect these cells with a receptor. And on one side, the outer side of the receptor, is a specific binding domain for something or an antigen which is overexpressed in the tumor cell. So it's like the antibody concept, okay? So it recognizes an antigen which is overexpressed on these tumor cells. That is obviously what allows the T cell to bind and find the tumor cells. The internal region of the receptor is really a T cell signaling domain, which allows the T cell to be activated. So it keeps the T cell in an activated state. So when you take them out into a tissue culture dish, you then transfect them, do that in a different way. You transfect them with these receptors, you multiply them, and then you inject them back into the patient. Now, one of the biggest obstacles that has to be overcome in this is really because you're relying on these cells to continue proliferating once you have injected them back into the patient, which can sometimes be one of the issues. Okay, so actually this is a therapy which is used, right? So this is an approved therapy for some cancers. There is a lot of clinical trials to use this type of approach for treating cancers, many other different types of cancers. But where it is approved at the moment and where it can be used at the moment is in a childhood leukemia, so acute lymphoblastic leukemia. This is a childhood disease or childhood cancer, and 80% of children are cured from this disease by using very stringent and intensive chemotherapy. However, obviously in 20% there is relapse, and very often the relapse leads to or is the cause of death in these children. Now, acute lymphoblastic leukemia rises in B cells, so it's a disease of B cells. These B cells, or these lymphoma cells, express an antigen which is called CD19. Now, these CAR T cells, so these T cells are obviously therefore engineered to target CD19. So this is the antigen which these T cells are targeting. So they've got CD19 antibody on them. Another disease for which these T cells or T cell therapy is approved is in B cell lymphoma. Again, the target, the antigenic target is exactly the same. It is CD19. So B cells express CD19, and these T cells have been isolated for these patients, are genetically engineered to target that CD19. That is what the cells are recognizing. Now, what are the issues with this type of therapy? One of the biggest side effects in some patients, you see what is called a cytokine release syndrome. And it's really a novel inflammatory process, and it can be life-threatening. So that is one of the main side effects. So as I've already said, there is a lot of research at the moment. There's many clinical trials which are looking at using this type of therapy for a whole variety of different cancers, targeting obviously different antigens which are overexpressed in these. The last one we're going to talk about is cytokine therapy. So cytokines, we talked about them already, how cytokines are produced by many of the immune cells. But cytokines can obviously be produced through recombinant proteins as well. For treatment, interferons and interleukins are typically used. Now, we don't really understand an awful lot about the side effects, but also how beneficial this type of therapy actually is. However, interferon alpha has been approved for a treatment of a number of different cancers, melanoma, capsuloid sarcomas, and blood cancers. And it's just be an example shown here, where IL-2 is now approved for this treatment of metastatic kidney and melanomas, as you can see in the article here. So these cytokines basically can be very toxic to cells. Okay, so we are now on to the questions. But I have copied, did I copy the last one though? So I'm going to change the screen. No, I didn't actually copy the last one. Okay, so why don't we start with these questions here? So what I would like to do now is that I would like to go through all of the MCQ questions that we have had in the different lectures. However, I am not going to go through them. I am going to sit and listen to you guys while you go through them. So I need somebody, so we're going to have different volunteers now, who is going to read out the question, who is going to read out all the answers, and once that person has finished reading the question and the answers, you guys need to tell that person what the answer is. Does that make sense to everybody? Somebody needs to talk now. Hello? So, can I get a volunteer, please? Who would like to read out a question? Now, this is very important for you because you need to understand, you need to learn to say the words, you need to understand the words, and you all have, I'm sure many of you have not passed your IELTS yet. You all need to pass IELTS before you can come and do your final year in Scotland. Okay, so your English is very important for you. Your English is also going to be very important for your final marks and the degree that you're going to end up with. Okay, so you need to practice as much practice as you can possibly get, and it doesn't matter if you don't know a word or if you can't say the word right. You know, everybody's here to help each other. Okay, you should try and speak as much English with each other as you can. So, is somebody wanting to volunteer and read out the first question, which I am going to put up on the screen?
May I have a try?
Yes, thank you very much. You're very brave. Right, I'm putting it up on the screen now, okay? Okay. So you read out the question.
Which of the following is not an I don't know how to read it.
Immunotherapy.
Immunotherapy.
Yes, very good.
Positive transfer of the
Serum.
Serum.
Yes.
Injection of T cells. Chemotherapy. Treatment with radiolabeled antibodies. Vaccination.
Yes, very good. Okay, guys, so what's the answer here?
I think the answer is
C.
Yes. So the answer is C. Okay, who's doing? Are you doing the next one as well? You want to do one more? Yeah? You want to read this one as well?
yes.
Thank you.
Who is thought to be responsible for pioneering protection against infection using a vaccine? The answer is B.
Yes, so the answer is Edward Jenner. Okay, very good. You don't, just if anybody think that they would like to read it out, you don't need to tell us the answer. You can let everybody else give you the answer, okay? So if you want, this is just a practice at reading as well. You don't need to know the answer, okay? Okay, who's having a go at this one? Come on, someone. We have had one very brave girl here. Can we please have somebody else? Come on. Who's going to read this one for us? Xi Yuping. No?
Oh.
Are you going to do it?
Okay.
Okay, thank you very much.
What is special about CAR T cells? A. They can be bought at safe ways. B. They are humanist B cells mixed with T cells. C. They are T cells expressing a radio label. D. They are genetically engineered T cells that target a specific tumor antigen. E. They are specific T cells.
And very good. So just for you that don't know it, Safeway is a supermarket where you buy food here in the UK. Okay. So which one is the right answer, guys? Which one are we thinking it is?
D.
Who said D?
D.
Yes, somebody said D, so the answer is indeed D. It is genetically engineered cells which are targeting a specific tumor antigen. Okay, we're just going to jump over this one, and then I'm going to go back to these ones here. Yeah, can you see them? Right, are you going to do the next one as well, please? Can you do this one as well?
Yeah. Which is question 1. Which of the following statements is correct? A. Virus can be viewed using a light microscope. B. Bacteria are always intracellular. C. Fungi are always pathogenic. D. Parasites are always macroscopic. E. Bacteria have a short germination time for hours.
So which one is it?
E.
Yeah. Yeah. Okay, who's doing the next one?
I want to
Wu
go into
Hong
try.
Chen.
Yeah, hi. Okay,
Hi.
I will do this.
Okay, good.
Which of the following is bacterial disease? A. Malaria. B. Bookworm. Hookworm.
Hookworm,
C.
yeah, that's right.
Tuberculosis. And D. D is candidiasis.
Yeah, very good.
Candidiasis. And
Yeah.
D. cold. Yeah.
Yeah. So
And
which
the
one
answer-
is it?
Answer is C. Yeah.
Yeah, very good. So you can do the next one as well, okay?
Yes, I can. Which of the following statements is correct? A. A pathogen has to be unicellular. B. A pathogen does not cause disease. C. A pathogen has a uniform structure. D. A pathogen have a diverse structure. E. A pathogen must be intracellular. And the answer is C, I think.
The pathogen can have a diverse structure, that's correct.
Yeah.
That's right. Right, who's for the next one? Are you doing one more? Yes, last
let
one.
me give the chance to other people.
I
Thank you.
don't think it's really. Okay, let's see who's next. Louis Yi Sheng Yi. Who wants to have a go?
I have a question
Okay.
about question 3.
Okay.
The third one.
A pathogen has a uniform structure. Yeah?
Yes.
It does not have a uniform structure. A uniform means that they all have the same structure.
Oh, okay, I got it.
Yeah, so they have a diverse structure. They all have different structures. They don't all have the same structure.
Oh,
Uniform
the
means
question is the question is A B C D.
I know.
So strange.
I know. That's a mistake. It's more, but you still got the right answer, okay?
Okay.
Okay, so who wants to try the next one? Who's going to be brave?
I'd like to have a try.
That is Shiruri. Shiru. What's your name?
My name
Shiru.
张芷睿.
Shiri.
对
Okay.
Yes.
Thank you. Okay, on you go.
And question 4 is, which of the following is correct? Our immune response can only deal with A, any type of pathogen, B, only intracellular parasites, C, extracellular parasites, D, only parasites less than 12 centimeters long, E, only extracellular pathogens. And I think the answer is A.
Yeah, A. So our immune system can deal with all of these, okay?
Okay.
In different ways. So it deals with all of these in different ways. You going to try the next one as well?
Okay. Yes. Question 1 again. Which of the following statements is correct for a viral pathogen? A, it can be seen without using any other apparatus.
Apparatus.
Okay. B, it is made of a mixture of DNA and RNA. C, it is only found extracellularly. D, a viral particle is called a virion. E, it is only found intracellularly. And I think the answer is D.
It's D. So a viral particle is also called a virion, okay? It is not a mixture of DNA or RNA. It's either one or the other.
Okay.
Now, however, so we do know.
Excuse,
Sorry?
I have a question. I have a question about this
Okay.
program. I'm not sure about the Z. I think various reply the host cell to live. So I think it's intracellular.
So that's a good question, right? However, what I need you to think back on, so what happens, the virus infects the cell, it replicates, what then happens to the cell? What does the cell do once the virus has replicated and matured? The cell bursts.
To destroy.
Yes.
The cell bursts, and what is released?
Virus.
The new virus.
So all these virion particles
Pathogen.
are now released into the blood, and they go on to infect other cells. So that means you have virus intracellularly, but you also have virus extracellularly.
Oh, okay.
That's how the virus is spreading. So it makes sense now?
Yes, thank you.
Okay, good. Okay, you're going for the next one as well?
Yes.
Who's going to read this one? It was sang something. Can't remember the name. Sorry. Are you going to do this one as well?
Let me have a try.
Okay, good. On you go.
Which of the following is a disease not caused by a virus? A. Influenza. B. Malaria. C. HIV. D. Ebola. E. Marburg virus. This should choose B.
So what is malaria? It is not a virus. It is a...
It's Malaria is
Parasitic.
the bacteria. But it's
Parasitic.
Parasite.
parasite. Yes.
It is a parasite. So malaria is a parasite. Yeah. It's caused by a bite from a mosquito. Okay. The others are viruses. Okay, you want to do the next one as well?
Okay. Which of the following is not true for bacteria? A, they have hyphae. B, they can be used to produce useful products. C, they must replicate to survive. D, pathogenic bacteria can be found intracellularly and extracellularly in the infected individuals. E, they can be neutralized by antibodies. This choose A. They have hyphae.
Yeah, they don't have that. What cells have got that? Fungal cells have got. That's right. So they have got it. Now bacterias don't. Good. Right. Okay. So who wants to have a go at this one? This one is a difficult one, isn't it?
I would like to try it if you don't mind.
Great, excellent. On you go.
Hansen et al. 2017. G. Microbial effect of antibodies on survival of against Pseudomonas aeruginosa, gram-active opportunistic bacteria. Bacteria incubated with medium containing 20% serum from mice. Vaccinated with first one is nothing, control serum. Second one is OPRO. What's OPRO stand for? I don't know.
You don't. It doesn't actually matter in this particular case because you really need to look back at the paper. So these are different. These are mice which have been injected with different things to generate antibodies, and it has then been collected. But you don't really know what they are. So it's really just different treatments, right? Which they're looking at whether they are effective against this bacteria.
Yeah. And sorry. What?
So really what you now need to do, so if you look at the figures, yeah, there's 4 figures. So these are different strains of bacteria. So what you need to tell me first is, if you look at DM125 strain, does DM125 respond to any of these serums? So to any of these antibodies?
I think it's seldom response. It's just have a slight response on the OPRA F and other. And also,
so basically there is no response. Okay.
Yes.
And DM126, any response?
No. Also, it's very similar with DM125.
Yeah. So if you then look at the PaO1 and the PAG, it is slightly different. So which one is most effective at killing bacteria?
The combination of OPRA F, L, and flag B is the most effective, and the second effective is OPRA F plus OPRA L, and the last effective would be OPRA F only. And and others are just don't have very obviously effects.
Very good. So that's correct. The triple combination is really the best one, isn't it?
Yeah.
The triple combination. So upon F upon L and flagellin is the best one at killing this bacteria. Very good. Do you want to try the next one as well?
Yes. It's which of the following is not an example of barrier defense in animals? A, skin, B, secretion from skin glands, C, exoskeleton, D, mucus, E, antibodies.
So do we know which one it is?
I think it might be C.
You think E?
E.
Okay.
It is E. It's antibodies, and the reason for that it's not a barrier defense. So it's an internal defense. Yeah.
Yes. I see.
It's inside, and your the other ones are barrier defenses.
Okay. Thank you.
Okay, who wants to try the next one?
Animal.
Sanqui, is that it?
I have a question about last one.
All right.
What about C?
No, the answer is E, because E is circulating. This one is actually quite a difficult one because of because actually many of them are, so secretion from, well, skin glands is not, mucus is not. But the correct answer is antibodies, because it's internal.
Do you mind to explain something about the exoskeleton? Because is that the exoskeleton is similar to skin for
Yes.
insects?
Yeah.
Okay.
You're right. And in obviously exoskeleton in animals, for example, could be like shells and stuff. So it's really very much refers to animals. You'll see in animals, a lot of animals have got shells and that, so that protects you. So exoskeleton in this case is also very much to do with skins and things like that, that protects you against any entrance of any pathogens or any microorganisms. This question is actually a little bit misleading, because exoskeletons is typically something that you obviously see on animals. Okay.
What about fingernail? Is fingernail?
Nail, for example, is a good example. Yes. So it is an external thing. All of these are typically external. Antibodies are not. You need to be infected. So the microorganism needs to get through all of these other barriers before it can encounter any antibodies. You basically need to have the infection before the antibodies before you encounter any antibodies. Well, all of the others is preventing access of the microorganism. Okay?
Yes, yes. Thank you.
Okay, who wants to do the next one? Anybody fancy trying this one? Yeah? You don't need to know the answer. Who's going to try it? Come on. We still have a lot to go through.
I want to try it.
Oh, good. Thank you. On you go.
The question is, the bacteria that are found in muscle surface and that play a role in defending the body against the parasites infections are A. Neutrophils, B. Eosinophils, C. Basophils, D. Monocells, E. NK cells, natural killer cells.
Yeah, good. They're hard to say, those neutrophils, eosinophils, and basophils. Okay, so which one is it?
C.
B.
Eosinophils, yeah. So you can really go back and look at some of the diagrams that was in the lecture, and you can see where these cells are, what it's B, eosinophils. Right, next one, are you going to do the next one?
Yes. The vascular changes of inflammation. It leads to an increase in bacterial cells at the injury site. B. It decreases the number of electrocytes at the injury site.
Leukocytes.
Leukocytes.
Good.
C. It allows plasma protein to move easily from the bloodstream to the injury site. D. It increases the number of antibiotics at the injury site. E. Activate lymphocytes.
Lymphocytes, yes. So what's the answer to this one?
I think it's C.
Yeah, it's C. You want to do the next one?
Okay. Let others do.
You do this one, then we'll let somebody do the next one.
Okay.
Okay.
Which is the criteria regarding acquired immunity? A. Acquired immunity only requires the present of helper T cells to function properly. B. Acquired immunity doesn't require exposure to a foreign substance. And C. Acquired immunity is triggered by contact with a particular antigen. And D. Acquired immunity includes inflammation. And E. All of the above are correct. And I think the correct answer is C.
Acquired immunity is triggered by contact with a particular antigen, and that is correct. Who wants to have a go at the next one?
May I have a try?
Yes, thank you. You have a go.
Memory B cells are A. Cloned lymphocytes that are active in subsequent infections. B. Cloned lymphocytes that are active during a primary infection. C. NK cells that recognize cancer cells and destroy them. D. Cells that produce antibody. E. Macrophages that have recognized self-antigen. And I think the answer is A.
That's right. So it's cloned lymphocytes that are active in subsequent infections. Does everybody remember what cloned means? It just
Replicate.
really, sorry. Please, yes, what did you say? What does cloned mean? Okay.
Replicate the
It's what?
maybe replicate the same sense.
It's the same, so it's against a specific antigen, yeah? It's cloned against a specific antigen. Are you going to try the next one as well?
Yes. The immunoglobulin that is passed from mother to fetus across the placenta is A. IgA, B. IgD, C. IgE, D. IgG, E. IgM. The answer is A.
D.
D.
D, the answer. You can actually find that on one of the slides. So some of the slides are showing which of the different classes of the immunoglobulins or antibodies can cross, because some of them can also cross the placenta, but to tell you the truth, I can't remember from the top of my head exactly which one of these ones or these classes that can actually cross the placenta. But when we are thinking in terms of breast milk, so obviously the ones which can be passed through breast milk is what we talked about in terms of gives the baby some passive immunity. So it means that the baby can be or have immunity to some diseases because some of these antibodies can be passed through breast milk. But remember that it doesn't give you long-term immunity. It's only short-term immunity because baby doesn't have any memory B cells against whatever this antibody gives you immunity against, okay? Oh, who's going to try the next one? Who's going to do the next three? No one?
Maybe I can try again.
You can try again. Good. So you're going to do the next three, okay?
Okay.
On you go.
The region of an antibody that is the antigen binding site is A. The constant region, B. The variable region, C. The heavy chain, D. The light chain, E. The hinge chain, the hinge region.
So which one is it?
I think the answer is B.
It's the variable region, exactly, because it has to, that's the one that is specific to each antigen. Next one.
A major difference between the activation of B cells and T cells is that T cells must interact with antigens bound to plasma membranes. B cells interact only with free antigens. C. B cells are not regulated by helper T cells. D. T cells produce antibodies. E. None of the above. I think the answer is A.
T cells must interact with antigens bound to plasma membranes. That is correct. Next one. You can do this one as well, remember? The next, please.
Cells that process foreign proteins and complex them with their MHC proteins are called, I think the answer is A, but I'm not sure.
Cytotoxic T cells.
Is it right?
Well, so it is not correct.
Steve.
It's antigen presenting cells. Because remember, the cytotoxic C cells can recognize MHC1 complexes. So all cells have got an MHC1 complex. And that MHC1 complex can complex with antigen. So if there is foreign antigen expressed in the cell, this will complex with the cell's MHC1 protein complex. And the cell will present that. And the cells which can recognize MHC1 is cytotoxic T cells. And antigen presenting cells, remember they have got MHC2 complex. And in the endosomes, so when these antigen presenting cells, these are also phagocytotic cells. So they phagocytose the microorganism, breaks it down, and part of those fragments can then complex with these cells MHC2. It's then transferred to the membrane, and it is displayed on the membrane of these antigen presenting cells. And then remember, the cell which can recognize these MHC2 complexes is what cell? What cells are recognizing this MHC2 complex?
I think it's hyper T cells.
So helper T cells, that's correct. So the helper T cells is recognizing that antigen. And when the helper T cells recognize the same antigen as the antigen which this cytotoxic T cell has bound to on the infected cell, the helper T cell then helps activating the cytotoxic T cell. Okay?
Okay.
Okay, we've got one left only, so do you want to just do the last one, or is somebody else desperate at having a go? Somebody really wanting a go at the last one?
Me.
Okay, you have a go.
Yes. HIV cause the toxic T cells. B. Preferentially destroys helper T cells that regulate the immune system. C. Directly inactivates plasma cells. D. Causes mutations that lead to autoimmune diseases. E. Does all of the above. And I think maybe B.
B is the answer, yes. So this is the helper T cells which gets infected by this virus. And eventually they're not replaced to the same level as in people which are not infected with the virus, and that obviously leads to a compromised immune system. Okay, everybody, so well done, and I'm happy that some of you know what you're doing. Yeah?
Yes.
So preparing a 5-minute presentation on that essay you wrote on BM110. So you've already had feedback. Remember to look at your feedback for the essays, yeah? And then make a 5-minute presentation on it. Okay?
Okay.
Try not to just write notes and read from the notes, because obviously can't see if you're reading from notes. Okay, so I will definitely see you for the presentation. So all these, what you also need to remember, so all these presentations are going to be marked. Yeah, you'll get a mark for them. So in the things that Eddie sent you, there is a marking guide. Did you see the marking guide? Yeah? There was like a table, and it tells you how much all of those different things are counting towards your mark. Now, it's very important you look at that one, because you need to make sure that you're fulfilling every area of that marking criteria. Because what you will get back, so when you're giving your presentations, I have to mark each of you. And I have to mark each of you in each of those areas. I might actually send you a more thorough marking guide so you can see exactly how we then have to mark, because you will get this back. So that is all uploaded on my place, and you will see that I have had to click for each of those criteria. I give you a mark from 1 to 9, okay? So obviously 9 is the best. It's actually 1 to 10. So for each of those criteria, I am marking you 1 to 10. Okay? And to get a 7 or an 8 is called excellent. To get a 9 or a 10 is outstanding. And to get a 5 or a 6 is average. Okay? I'll maybe actually forward you those so you can see them, exactly how the marking is done, because you will be getting that return. So that's what you can see on my place. Okay? For each of those marking criteria, you can see what score you got in your presentation. Okay? So I will definitely see you all for your presentations. But I think Eddie and I might be planning to do a workshop together before that. So I might speak with you all before then, okay? Otherwise, have a lovely weekend. It's Friday. So you have 2 days of rest now, okay? So enjoy your weekend, everybody. And I'll speak with you all soon, okay? Bye-bye.
Bye.
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 Bye.